Welcome to the workshop and welcome to Institute of the Mathematical Sciences, Chennai. For those of you who are from outside, also welcome to Chennai itself. So we are sorry we're running a bit late, but the inauguration is actually going to be pretty short, so hopefully we'll be back on schedule in no time. I just wanted to make a few quick announcements. The name tags are not, weren't actually given along with the registration kit, but they're here now. So we'll have another short round of registration session for those who are arriving late between 11:15 and 11:45. So please pick up your name tags during that time. So during the next T session that we have, which is between 11:15 and 11:45, please pick up your name tags. I'd also like to point out the two volunteers we have in IMSC. Anand, yeah, so can you just stand up and raise your hand? And Janaki? Yeah. So, if you need any assistance, you could either contact me or you could contact Janaki or Anand. And especially if you are, giving a poster and so on, Anand and Janaki would coordinate the poster sessions. As regards internet, so we do have Wi-Fi connectivity here. So I'll be trying to see if we could get just a INCF account for all of you. So you don't have to create your own accounts for the Wi-Fi individual accounts. So those of you who are staying in IMSC guest house, of course, might have already created your own Wi-Fi account. So that's good here. Otherwise, we'll try to see if we could get just an INCF specific account. Okay, and important things like lunch, tea, coffee, dinner the times are already mentioned in your schedule that is given along with the registration kit. Yeah, so that's it for now, and now I'll give the mic over to Upi to say a few things about the INI and INCF.
And thanks to Srini, who's around here also. So Srini and Sitabro are the co-organizers of this event, and this is the third in the series of Indian node events on behalf of, this is the third of the series of workshops for the INCF that has been organized by the Indian node. First one was in Bangalore, which is when the Indian node was formalized. Second one was last year in NBRC at Manesar, and this is the third. Along the way, we've also had summer courses, the most recent of which was actually organized by Kaushik over there this summer. So that was on neuroinformatics and computational neuroscience. So the INI stands for Indian National Node for Neuroinformatics, and it is one of the many national nodes of the INCF. And Sten Grillner here, who's our first speaker, is the chair of the INCF for another few months. So our job as INI and as part of the INCF is to promote neuroinformatics and computational neuroscience type science, research, technology, and all the other good things that flow from these. And so, ours is the Indian node is a new one, and so but we've started, I think, reasonably well. We have some 22 members now, and every year 1 or 2 new institutions are joining the node. This meeting is our flagship. It goes from one member to the other every year. And those of you who are node members are keenly encouraged to think about hosting something like this at your place in a future year. So we're all here to hear about some excellent neuroinformatics and computational neuroscience. And with that, I welcome all of you and hand it over to Sten.
So it's a great pleasure and privilege to be here and to see how well the Indian node of the International Neuroinformatics Coordinating Facility is developing over now a few years, 3-4 years, something like that. And with that, I will change over to science and talk about the logics of networks in motion and from ion channels to behavior. Interesting. Yes, no. So the motor system has a motor infrastructure with a number of neuronal networks that are preformed to coordinate different movements. You have in the spinal cord, you have the networks underlying locomotion and protective reflexes. In the brainstem, you have the networks underlying breathing and chewing and so forth. One can illustrate that by saying that this is a spinal network when it is initiated. When activity is initiated from the brainstem and you have the networks in the spinal cord, when you initiate the activity, locomotion will start. If you happen to be a fish, you will start swimming, and you have the networks underlying respiration in the brainstem, which of course operate from the day you are born to your last day. You have the networks underlying chewing when you turn them on. You will chew. Chewing is a mammalian invention. Crocodiles and the like, they bite and swallow. We have networks underlying saccadic eye movements and orienting movements. When you stimulate a given point in the superior colliculus, you will initiate a saccadic eye movement with a given direction and a given amplitude. So you have like a motor map that is used for steering eye movements and the subsequent movements of head and body. And you have the different networks underlying expression of emotions. The emotion of this guy, I think, is pretty clear to you. Humans have 7 different distinct motor programs for expressing different types of emotions. These are all coordinated at the lower level below the cortex. Cortex in humans is involved particularly for fine motor skills like speech and hand-finger control, like when you play the piano and so forth, which is of course, they also modify ongoing movements and reaching. Essentially, you have a range from very simple protective reflexes, over swallowing, respiration, locomotion, posture networks, chewing, expression of emotions, the eye motor map. You could also include reaching there. So having this number of different networks, and it's a much larger network, that's the motor infrastructure that we are born with and which develops, and of course can be refined through learning, adapted through learning. But it's a basic infrastructure that you have. These networks, of course, having them like this, they also need to be controlled. And their selection of behavior, sequencing, and timing is of critical importance. And in that, the basal ganglia plays a very important role, as you will see later. I will talk essentially a little bit about how these networks are designed and how we have used modeling to explore them, and also the mechanism underlying selection. This sort of organization is essentially very conserved. The same type of control occurs in lamprey and fish, as well as in mammals and primates. So I will talk about locomotion, as I said, and it's organized again in a very conservative way. You have the networks in the spinal cord from lamprey to man that generates the basic pattern of coordination and movements. As an integrate part of that, also sensory feedback plays an important role, but this is sort of the essential pattern generating circuitry. You have the locomotor command regions in mesencephalon and diencephalon, which can be stimulated, and you can induce locomotor activity. If you stimulate the region, this region in a fish or a lamprey, it will start swimming, and the more you stimulate, the more fibers you involve the faster it will swim. If you're stimulating a bird, it will start walking, and if you increase the stimulation, it will start flapping the wings and trying to fly. And in a mammal, you can go from slow walking to trot to gallop, if it's an animal that gallops. So essentially, this is a very useful and simplifying control strategy. A tonic signal from these command regions can activate the pattern generating circuitry in the spinal cord that also can change from walk to trot to gallop, so by interaction of the different CPGs for the different limbs. So this is essentially a propulsive synergy. If you will then, of course, to have goal-directed behavior, you need to steer the movements, and there, at least in the Lamprey, tecton plays a major role, and also what you need to have a control of body orientation posture. Having these possibilities, you have what is needed for goal-directed behavior, not just propulsion, and then you need to have a mechanism to select, to determine when locomotion should be generated. That's where the basic ganglia play a role. I will talk primarily about this and about this, which is our more recent work. So what we decided now quite a few years ago was that mammals were rather complex, and we needed to have a simpler model. So we choose as simple a vertebrate as possible, but still a vertebrate with the same basic design. So instead of many of our fellow researchers work on this and try to infer things about this guy, perhaps even that guy sometimes, we are inferring from this level to this and sometimes this. So we selected the lamprey, which is a cyclostome, more primitive than an ordinary fish, no jaws. And it diverged from the main vertebrate line very early on, for 560 million years or so. But it still has all basic features of the vertebrate nervous system developed. So that's why we did that. And now, many years ago, we have outlined the system, defined which neurons are active, used paired recordings, etc. So we have recorded from the pre- and the postsynaptic cells to identify the types of neurons we have, their synaptic connectivity, synaptic properties, and so forth. And essentially, in the lamprey, which swims with alternating movements, we have the excitatory interneurons, populations of them that synaptically interact, and they can generate the burst activity all by themselves. They are coordinated by inhibitory glycinergic neurons which cross, and they both then project onto motor neurons that generate the motor pattern. We also have GABAergic interneurons that contribute to basic presynaptic inhibition on the sensory afferents and also on the premotor interneurons. So these are the basic players. And formulated in a slightly different way, we have in each of the 100 segments, we have excitatory interneurons, about 40, 50 or so, that synaptically interact to some degree. And these, when you have isolated out all inhibition, they can still generate the burst pattern, sort of bursting, so they provide burst activity. The coordination between the two sides is then via inhibitory interneurons. So essentially, the excitatory core of spinal glutamate neurons generate the burst activity, and the inhibitory neurons, under these conditions, generate the alternating activity. So that's the essential thing. Of course, it's what makes these bursting. When they are turned on from the brainstem, that they would fire, it's clear, but why would they burst? Where there the membrane properties come into play. You have these different neurons. Of course, they generate the action potentials, But when activated, they activate both NMDA receptors and also voltage-dependent calcium channels will be activated. You have an accumulation of potassium of calcium in the cell that activates calcium-dependent potassium channels, which will pull the membrane potential down again. We showed some years ago that you also have, during high level of activity, sodium-activated potassium channels that will also provide hyperpolarization. So you have the addition between these two players that terminate the burst activity. So essentially the burst pattern is interaction between excited interneurons and membrane properties of these cells. Not in the isolated nervous system can generate this pattern, but in real life you also have sensory control. You have stretch receptors that are activated during the alternating movements. And if, for instance, this side is active, the stretch receptors on this side will be extended, and we have blue inhibitory interneurons that feed back information to inhibit these group of cells to help terminate the activity, and we have correspondingly ipsilateral excitatory stretch receptors which will promote activity like this. So there's a sensory overlay that helps coordinate the activity under normal conditions. Then we have the descending control from the brainstem by the reticulospinal neurons that turn this on, and they also are rhythmically active because information is fed back to this level. And then we have the locomotor command regions, which I spoke of, which normally turn them on, and on the top of this we have the basal ganglion. Now what I've mentioned so far is this synaptic interaction via ionotropic ion channels, like NMDA and AMPA on the excitatory part, like glycine on the inhibitory part. In addition, we have, of course, the metabotropic receptors that also do not provide control from cycle to cycle, but provide the fine-tuning of the network. And so we have 5-HT neurons are turned on and modulate the activity a little bit. We have a little bit of dopamine, GABA B, and tachykinins. We sort of modulate the network activity. I will not speak more about this. I could have given a separate lecture on that, but I will not. But that's other important players, and if you think about spinal cord injury, etc., these are also important players to consider. So I provided then a very general description. What was very important early on in this analysis was also to test this, and then we tested that through with computational methods together with Anders Lansner and Örjan Eckerberg and also Jeanette Hellgren that will hear speak later on. So essentially we simulated each type of interneuron that we had defined with their appropriate properties as close as we could to their natural counterparts. So they had the after hyperpolarization, the different phases here. They had the same response during repetitive activity. We simulated the EPSP, both the voltage-dependent NMDA receptors and the AMPA receptors. This is now quite some time ago. And we had initially 5 compartment models of cell body, initial segment, and 3 dendritic compartments. Lately, we have used rather many more compartments. And we simulated with a sort of Hodgkin-Huxley neurons with sodium, potassium, calcium-dependent potassium, sodium-dependent potassium, the 2 different types of calcium channels. And when we did that, we could show that if you activate reticulospinal neurons, you then turn on the network generating alternating activity, the inhibitory and excitatory on one side, and their interaction. Moreover, what was important also was that we could drive the network from slow swimming, about 0.5 hertz or so, up to 10 hertz, which is a natural frequency range of this network in the living creature. So that's for one segment. Of course, there is not one segment, but there's 100 segments. And essentially, when the lamprey swims, there's a wave of activity along the body. There's a phase lag that is constant, regardless of the cycle duration. This same phase lag can be generated also in the isolated spinal cord, a little bit more variable, but essentially it's the same values. So the spinal cord is hooked up to generate this pattern. But if the lamprey is caught in a corner, it can swim backwards for a couple of cycles, and if you do that, you reverse the phase coupling. One can also reverse the phase coupling experimentally by providing extra excitation here or extra inhibition here, as I will show you in a short while. We have then done large-scale simulation of the intersegmental coordination using a Blue Gene supercomputer, and this is very much together with Jeanette Hellgren and Alex Koslov and Anders Landster to some degree. And so we have had the approximate correct number of cells with 100 cells in each segment, developed by Huss et al., and a fair number of synapses. So essentially, these neurons have cell body, and they have two dendrites, and then you have further ramifications. And the advantage of that is that you can have input to apply to different dendrites, not that we have used that very much. And what is important is that we have then also introduced the differences in size that you see experimentally, and also the changes in other properties, the normal variation of within each category of cells. So some cells, if you inject current to them, some cells will start respond with a very low rate of activity, others with a much higher rate of activity. And this variability in cell properties turns out to be quite important, because it makes for a gradual recruitment, a gradual de-recruitment. And if you set the membrane properties the same, all the cells will respond in an identical way, and you will have a much less stable activity, etc. We have then used the connectivity with the excitatory interneurons having ipsilateral descending and ascending axons, and the inhibitory cross ones a bit longer, but essentially connected, as I said before. They had been displayed with a bluish color for hyperpolarization, red for not yet spiking. If we then look on these 10,000 neurons from above, this is head, this is tail. You can see that you have alternating activity of left and right that are propagated down, and they are propagated down with about, so it's about one cycle throughout the spinal cord. This corresponds to the normal, or the approximate normal number of cells. So we can, the network hooked up, as we find biologically, can indeed generate this pattern. Now, what about the flexibility? If we, for instance, modify the excitability of the most rostral part of the segments, can we change the pattern of activity. And if we provide inhibition there, as you can see, then these segments will have a lower excitability and tend to burst slower, and they are brought along by the other segments that has repercussions all the way. So essentially, you have a situation where the entire spinal cord is coordinated by modifying the excitability just with a tiny little bit of the spinal cord, the rostral-most segments. So essentially, if you have the coordination here of the excitatory neurons from head to tail, they provide the phase coupling that you see. If you add inhibition of these, you will have a progressive reversal of the phase coupling. If you instead add the excitation, which happens sometimes, then these segments will be the leading and the other will track. So you have essentially a very convenient type of control in that you can just tinker a little bit with the most rostral segments and thereby controlling the entire segment to make it be coordinated. So I think that's also a simplifying design. You do not need to have signals that go from the brain all the way down there. You can just play with that if you like, if you're a Lamprey brain. So then a generalizing thing about excitatory unit burst microcircuits. As I showed you, well, I told you rather, and showed you, that in locomotion, burst generation can occur without inhibitory interneurons. You can block all inhibition and you can still generate the burst activity. And this goes for Lamprey, it goes for tadpole, it goes for rodents, and it goes for cats. And most likely also for primates. If you look on respiration, you can actually block all inhibition also in the respiratory system and generate rhythmic activity. You have, of course, inhibition used for coordination, but the core excitation is really can be generated. So the core of the core is excitatory. Also in chewing, and also the saccadic burst generation, you have mutual excitation which provides a burst, normally followed by very efficient inhibition right away. But essentially, one can say then that you have different networks that can generate burst activity, and the coordination between burst generations, of course, burst generators is dependent on inhibition, with mutual excitation between the two sides of the Prebutzerian complex, for instance, or combinations, etc. But I think it's one important fact. And with that, I'm not saying that inhibition is not important. It's of course critically important, but it's a subject, but mainly for the coordination. We have then also used models now much earlier and now more recently also with neuromechanical models where we have the same type of network as I just mentioned, controlling muscle segments that are simulated with viscoelastic properties, etc., as Örjan Eckerberg pioneered and then Alex Koslov has been doing now, so that we can generate a fish that swims. We are also simulating the viscous properties of water, etc. And we've used that not only for locomotion forward, but also for steering in our more recent work of tectum and how tectum can be used to control orienting movements. So essentially, we are having a control of MLR, the command region which generates activation of the two sides, And then, in addition, we have signals from tectum that then control reticulospinal neurons on one side or the other and can elicit turning by that. So it has also been quite important to use this type of simulation. If I don't speak, it's fine. Anyhow, this is a simulated lamprey, and we can just have it swim like that. And this is now controlled by a similar network as we find, as we utilize. So this is forward swimming. You can also see that the movements are very much corresponding to the normal swimming movements. And then we have also analyzed in quite some detail the commands for steering. So essentially, this little guy here can swim, and it can be made with a signal from tectum to turn again. So with that.
Cell phone, use cell phone.
Cell phone? No cell phone. Good. So now I will switch over from to goal-directed motor behavior and selection of motor programs, and I will focus on the basal ganglia. Okay. So first, a very simplifying graph from mammals as well as lower vertebrates. The design and the output stage of the basal ganglia is pallidum and substantia nigra reticulata. They all contain GABAergic neurons that are tonically active. And subpopulations of these project to different parts of the brainstem, the command centers for locomotion, postural parts, saccadic eye movements, which was the first to be demonstrated by Hikusaka, and also chewing, swallowing, and so forth. So it seems that one has subpopulations of cells here which provide tonic inhibition and of the different brainstem command centers. So essentially, at rest, the function seems to be to have a tonic activity that keeps all these different motor programs under tonic inhibition. We also have signals fed back to thalamus and up to cortex. And I think in the next talk we will hear more about the output stage and the other. So then we have the other group, the input level stratum. And the input level here consists of subpopulations of cells that are thought to project directly to subpopulations here, which will then control motor behavior. Stratum is very different in that stratum consists of cells that are designed with inward rectifiers that tend to hyperpolarize the neurons if they don't have input. The normal input they have is from thalamus, glutamatergic, which is about almost 50% in rodents at least, and then from cortex. So essentially, these two inputs will determine whether these neurons that are difficult to activate will fire, and if they will fire, they will in turn provide inhibition here and remove inhibition here, and thereby induce a behavior. At least that's our interpretation of that. There's one more important player in this, and that's the dopamine input. The dopamine input, primarily to stratum, but not exclusively, is important. You all know that if you have no dopamine input. As in Parkinson's, you have severe problems of initiating movements, etc. Conversely, if you have too high levels of dopamine, which can happen as a side effect of medication and so forth, and also in other conditions, then instead of having difficult to initiate, you have spontaneously initiated movements. You have what's called hyperkinesias, movements that are initiated without the intention of the individual, which I think is also speaks to the importance of these circuits for control of behavior. Now, what happens if you remove one of these inputs? If you remove the entire cortex, and these are experiments that have been done over quite a few in the 50s, 60s, and 70s, essentially in rabbits, rodents, cats. If you remove neocortex, often early on in evolution, these cats will survive for years, and they have a rather complex behavior. So this is an example from a study from the 70s by Norcell, and essentially this cat, which has had neocortex removed, I think 2-3 weeks after birth, and then have grown up. And what is important is that neocortex was removed, but stratum and all other parts are still intact. These cats, this is a cat of this type being put on a box like this, knowing that there is border palpating for this. These cats can use vision, but they don't have a detailed vision. So essentially, they can also get hungry or thirsty. Then they search for food, and of course they eat. They can also remember the location of food in the T-maze. And they can find their way out of a complex maze. And finally, they can also display aggressive behavior towards other cats. If you have them in a colony of other cats, these cats will attack the other cats and successfully so the normal cats choose to avoid them. So essentially, the conclusion is that a very large part of the neuromachinery for goal-directed motor behavior is subcortical. You can initiate a lot of these activities. You can control them. And by that, I'm not saying that cortex is not important. Cortex is, of course, important for evaluating the conditions around them. And these cats seems to be, they have difficulties in making appropriate decisions. They then rather attack than stay back. But what it does mean is that cortex has a very clever infrastructure to work on during normal conditions that can handle most aspects of normal behavior. So I will then, what all of you know is the direct pathway now both in mammals and Lamprey, as I will show you. Stratum has a direct pathway which I just spoke of that has a large part which is controlling brainstem circuits and some fed back. What I did not tell you is that we have what's called the indirect pathway with globus pallidus externa, which Jäger will speak about in a short while, and the subthalamic nucleus. You have bidirectional activity here and also there. But this indirect pathway is another important player. You have the input from thalamus, you have the input from motor pallium in this case, and so forth. What I will show you is that all these circuitry, which was thought to be primarily mammalian, is present in the lamprey. So this circuit existed very early on in development. I should not say that the direct pathway is controlled by a subset of medium spiny projection neurons with D1 receptors, and this pathway is primarily via D2 enkephalin. So different subpopulation of cells controlling the two different parts. So why should one do this in lamprey when so much work has been going into the rodents? Well, we have the very limited behavior repertoire, and that is an advantage. The lamprey is sort of stupid in that it has a limited behavior repertoire. Rodents and mammals, of course, have much more. We have comparatively few neurons. We have a detailed knowledge of downstream command centers, which is not quite true in others. And we have the evolutionary considerations, which I think are quite important in themselves. So the lamprey stratum, we have recently reported, and these are Jesper Eriksson, Marcus Stevenson Jones, and Britta Robertson that has been doing a lot of work on this. What we have shown is that the stratum has the same type of input, histamine, dopamine, 5-HT, going on to stratum and also from cortex, pallium, and thalamus. We have neurons that are spiny, the projection neurons, as much as the basal ganglion neurons. We have the markers, the gene expression. These neurons are GABAergic. There are some cholinergic. There are the D1 substance P ones and the D2 enkephalin ones, and we have the inward rectifier neurons also. So we have the identical markers as we have in our own brain. We have the dopamine denervation. If we kill off the dopamine neurons with MPTP, we have a very much reduced locomotor activity, difficult to initiate activity, and a lot of the other characteristics of Parkinson's. And of course MPTP is a rather dirty drug, so one can essentially then test whether it is the dopamine denervation that is special, and that's with apomorphine, a dopamine agonist, and then you recover function. So this is the same type of symptoms as we have in mammals. These neurons are have inward rectifiers when you inject. So we record patched these stratum neurons in slices from lamprey. We could show that they have inward rectifiers, so these channels are open at rest, providing hyperpolarization. When you depolarize, they are closed. So to make it a long story short, we have these two subtypes of dopamine projection neurons with D1 and D2 receptors. We have the input, we have dopamine denervation, it leads to hyperkinesia. We have the medium spiny with inward rectifiers. And then what I have not told you about, and what we are just publishing, is that the characteristics of the synaptic input from thalamus and from pallium corresponding to cortex is that we have depressing excitatory synapses. The first EPSP is much larger from thalamus, and from pallium they summate nicely to our facilitating synapses. And that's the same finding that recently has made in rodents also. So that speaks to that this is a fundamental property. So essentially striatal structure and function is conserved throughout vertebrate evolution. Now is there the output level? When we started this, it was claimed in the literature that there was no output of the basal ganglia, but we could show that globus pallidus interna and substantia nigra reticulata exists. So essentially, one globus pallidus, we knew that we had GABAergic projection neurons there, we could show that stratum has an input to them. When we recorded from them, we could show that they were tonically active, and if we blocked the thaumaturgic synaptic transmission, they still remained active. And this is these neurons and no neurons around this area. So they have the same tonic activity. We could show that we have separate projection neurons from the different areas that provide output. So there is a direct pathway. We have the globus pallidus interna. We have these. We have the tonic activity. They are parvalbumin expressing, which is a characteristic also in mammals. And we have the D1 substance P that projects directly to the output level. I have not shown you that because lack of time. And then does it have an indirect pathway? I will not show you the slides. I will just summarize that and say that the stratum D2 receptor expressing neurons with enkephalin, they provide to what corresponds to globus pallidus externa neurons, which in turn project to the subthalamic area, and the subthalamic area projects back to pallidum. And they are spontaneous active, they are post-inhibitory rebound. So again, we have all factors. So essentially, we have stratum, we have this, these neurons, we have the projection there, we have this. I don't have the arrows back there either. But so essentially, this part is conserved, and we have also shown that you have, I will not show you that, but there is the myotripallium that projects here and projects there, which is sometimes referred to as a hyperdirect pathway. Now to test, we knew then that we have tonic activity in these, we know that they projected to different motor centers in the brainstem. So the prediction then is that if we blocked this tonic inhibition, we would be able to release behavior. And we tested that first by injecting GABA blockers into MLR to block the effect of the spontaneous activity. If we did that, we then released locomotor activity with alternating activity, as you can see. If one has it in a semi-intact prep, we also release the swimming motor behavior. And we have done the same type on the other motor centers for eye movement, etc. So essentially, the net effect is a tonic inhibition that is effective behaviorally. If one does the corresponding here of stratum, instead of stimulating stratum, thereby inhibiting pallidum, thereby removing inhibition from these, one can also elicit activity. And what we did here was to record from the reticulospinals that provide a command to the spinal cord. When we do that, we get a strong depolarization, and with spiking, and at the spinal level, we then get rhythmic activity after a while that generates rhythmic activity. Again, confirming that we can elicit this pattern of behavior. So essentially, what we have shown is that we have, in mammals, we have stratum, pallidum, and an indirect pathway. Also in birds, it has been recently shown. We've shown now that at the lamprey that diverged from the main vertebrate line some 560 million years, we also have this organization, which means most likely that very early on in vertebrate evolution, this type of command system was invented, and it has been retained throughout vertebrate evolution. And I believe that if one tested these different groups, one would be able to find that. So let me just say that, how do we think about the control? as a conceptual scheme. We think of modules controlling each pattern of behavior, like locomotion, eye movement, steering, feeding, and we have the different components like the GPI, GPE, etc. in a sort of module. How they project, one may guess about. The direct or indirect pathway responds to the input from pallium and cortex. And what has happened during evolution is that one has probably added progressively more and more modules to control a more and more complex pattern of behavior. That's our working hypothesis. So the stratopallial circuits, you can see the basic ganglion circuits for gating and modulation of basic behavior have been conserved from cyclostomes to primates. And these circuits probably arose in cyclostomes to control basic patterns of motor behavior, like locomotion and feeding. And during evolution, this modular circuits appears to have been hijacked to control more refined motor patterns like independent finger movements and possibly cognitive functions. Let me just finish showing that we have also worked extensively, or is in the process of working extensively and in incorporating the basal ganglia in the simulations. So we have incorporated here a stratum with cellular properties, a pallidum, and an MLR, reticulospinal level. And here you see the spinal cord again, and if we do this, we can see that stratum is silent, pallidum is firing along, some activity in the reticulospinal here also. When we activate this, then this is turned off, this enhances its activity, and you can see that things starting to happen here. And if you are a little bit patient, you will see that this has initiated activity. So that's a proof of principle. We have also simulated this with activation of MLR, drive of the reticulospinal to CPG, and this is sort of exploratory simulations, I would say, but also that we have subpopulations for turning left and right, and these when you activate one like this, you will then turn on this, which will provide extra excitation on the two sides. And if you have symmetric activity like this in stratum, if you then enhance activity in the left or the right subpopulation here, you will then inhibit pallidum, thereby eliciting turning responses. We have done that, and this is just an example where, and this is more fiction, but these are with Kostev and Eckerberg, and essentially having a little bit of vision, and they can swim towards blue object and avoid red object. And as you see here, this is the little lamprey swimming, little model lamprey swimming, and you can see that it goes for the blue object. Now, this is exploratory simulations, but of some interest. So essentially what I've been trying to tell you is that the Lamprey motor system from forebrain to spinal cord is in some degrees like a blueprint of the mammalian system. I have compared it now quite a few times as a Ford Model T versus a Ferrari or a Lamborghini or whatever you may choose. So essentially, the Lamprey invented the basic ganglia control already at this point. Mammals, which are relative newcomers, came here, and humans out of proportion here. So it seems that this is a very conserved machinery for the control. And with that, I will finish and just thank my collaborators in this work of the basal ganglia. It's very much Rita Robertson and Marcus Jones, Silverberg, Ericsson, and also the interaction with Jeanette that has been important in the modeling, and Alex Kostler in particular. And for the spinal work, which is the more older work, Peter Wallén has been of particular importance, and a number of other colleagues. And how should we like to pay tribute to this guy? Okay, thank you.
Just wondering, see, a lot of people have been developing a lot of actor-critic models of basal ganglia function.
What did you say?
There's a lot of effort to develop actor-critic models of
Actor-critic models.
So is it possible to apply those concepts to
It's
Lamprey?
possible to do that in, no, we have not done that so far, but I mean, that's, what I've talked about so far is a tonic input of the dopamine. You then have, which I could have talked about also, the dopaminergic reward-related input, which relates to habenula, and also the control of the habenula and thereby the dopaminergic thing. That's also conserved in great detail. So we've done that. But we have not modeled that, but that's of course of great interest to do.
Yes, very nice talk. I was interested, so you have all these parallel pathways of different movements being selected. It seems that you would also need to crosstalk between all the different pathways to only select one movement and have them coordinated correctly. Where do you see that taking place?
I mean, what we have, if you think about the indirect pathway could be used to either just to break a given motor pattern, like locomotion. You initiate locomotion, I direct, and you but as you indicated, you of course need to prevent a number of other movements, and the indirect pathway can also be thought of to inhibit the different motor patterns that are not compatible with this. I mean, some motor patterns are compatible. You can walk and chew at the same time, at least if you're not pressing forward, as I said once upon a time. But you cannot turn right and left at the same time. You could have you need to have one decide for one or the other. You could have that decided at the lower level, or you could have that decided at the basal ganglia level. But it's interesting.
Part also induces rhythmic motions, especially in rodents. They induce rhythmic motions in the hind legs.
If you inject
Dopamine directly onto the spinal cord. Do you think there might be direct dopamine projections onto the spinal cord?
There are in rodents at least a direct dopamine projection, a small but described by Anders Björklund many years ago. Of course, the spinal cord network is normally activated by glutamatergic pathways, and in practically all vertebrates, 5-HT pathways also contribute importantly and elevate the excitability. And the 5-HT pathways are probably more important in the fine-tuning of the network, so it starts operating. We showed that in the cat many years ago, and that seems to be the lesson learned everywhere. So I think one thought with spinal cord injury is, of course, that if one has a partial lesion but is wheelchair bound, then either with electric stimulation of the cord, which is there are techniques developed, one can perhaps promote the functional circuits a little bit better, and also with intrathecal injection of precursors of, for instance, like dopamine dopamine and noradrenaline, which is a more important part there, or parameter. So it's a lot of things one can do, I think, combined with training especially.
Okay, so thank you very much, Sven.